And then we'll have our next speaker for this session, Elena Rondeau, also from the ImmunoConcept lab that has been mentioned a few times by now. And she will be presenting on a similarly topic, also about cancer progression and its control. And we are very much looking forward to hearing your talk.
Hello, everyone. Thanks a lot, Gregor, for the introduction. So I will open my presentation. Can everyone see and hear me?
Yes, looks and sounds good.
Okay, perfect. Thanks. So yeah, thanks a lot for giving me the opportunity to talk during this wonderful workshop. So I'm from the ImmunoConcept lab in Bordeaux, as Gregor mentioned and Martin talked about a few times. Currently doing my PhD there with the main question of better understanding cancer metastasis and its control, with a particular focus on the implication of the immune system. So I am working on an interdisciplinary project with some work in the conceptual immunology team with Thomas Pradeux, and some lab work in the cancer immunology team at the same lab. So my presentation will be separated into three main parts: a short introduction, a more detailed part on my research project, and a few ideas for discussion. So as I said, my main question focuses on better understanding the process of metastasis, which corresponds to the migration of cells from the primary tumor to a distant site. For example, here from an epithelial primary tumor to the lung tissue. So this process of cancer progression is the most deadly aspect of it, as mortality increases with the development of secondary tumors in distant sites. It is often resistant to conventional therapy targeted at the primary tumor, and it remains challenging both clinically and conceptually. So usually it is described as a succession of steps that comprise the invasion metastasis cascade, from the proliferation of cells in the primary tumor, mobilization of a vascular network, the ability of primary tumor cells to detach from the primary tumor and invade local tissues, then go through the circulation, blood or lymph, and finally, when they arrive at certain sites, extravasate and potentially proliferate if the secondary milieu is favorable. So this illustrates a significant shift of our focus in research and therapy from the local tumor microenvironment at the primary site to the inclusion of host components at the organism level, so what we may call the host macro environment when secondary sites are affected. So when trying to conceptualize the different steps of metastasis and their causes, we have thought about the succession of steps and the pro-metastatic processes that are involved, from the promotion of exit from the primary tumor, then efficient circulation and attraction to the secondary site, and trying to distinguish tumor cell intrinsic causes that may act at each step, such as genetic characteristics or local interactions with the different microenvironments, from tumor cell extrinsic causes that include other non-tumor cell components, such as vascular elements, circulating elements, or components of the pre-metastatic niche termed here PMN. So the different causal links that may exist between the factors and the processes may be of different kinds. So this is an attempt to think about these different links. So for each step, 1, 2, and 3, we have tried to describe what could happen between the factors at a given step on the process. For example, for circulation, resistance to cell death for a tumor cell intrinsic cause, or aid from vascular elements that may protect the cells in the circulation for an extrinsic cause. But at this same step, some influence may come from the previous step, as in B. For example, the promotion of epithelial to mesenchymal transition promotes the ability of cells to circulate, or some elements of the extracellular matrix may influence the ability to circulate. And the influence may also come from a more late step in the process. For example, stable interactions with the future secondary milieu, or interactions with some components that are present at the secondary site and that may come from the primary tumor environment. My main focus for the rest of the talk will be on the secondary site, and more specifically on the notion of pre-metastatic niche, which is the idea that once a primary tumor has proliferated and tumor cells exit the primary site, these may establish efficient residency in secondary organs. And in these organs, an interaction exists both with local components such as fibroblast or immune cells, and with components that have been mobilized because of the development of the primary tumor, such as the case with bone marrow-derived cells after signaling from primary tumor-derived elements. So this shows the importance of the host components mobilized in the process, and also push us to ask how specific this remodeling is depending on the secondary site. So this is actually the main core of my research project, which is interested in tissue tropism or secondary organ specificity in metastasis. So this idea of organ specificity corresponds to the fact that depending on the type of primary tumor, only certain secondary organs will be affected. So for example, here, breast or prostate cancer often develop metastasis in the bone or lung, whereas abdominal cancers may affect the liver. This was first described so in the late 19th century, in 1889, in a clinical report by a British surgeon, Stephen Paget, who analyzed breast and other types of cancer patient autopsies and assessed the number of patients affected by metastasis at different secondary sites. So he realized that depending on the type of cancer, certain secondary organs were more affected than others, with a particular focus on breast cancer and the existence of liver metastasis. So he proposed that the idea that one remote organ is more prone to be the seat of secondary growth than another, which would mean that the disease has its seats of election. So the question here was, what decides which organs suffer in disseminated cancer, with different possible hypotheses concerning secondary organ predisposition or diminished resistance in non-affected organs. The main proposal is that these remote organs cannot be altogether passive or indifferent as regards embolism. And so the challenge is to understand the connection between the primary tumor and the derived secondary growth. In order to illustrate and conceptualize this notion, Paget proposed the seed and soil analogy by using botanical comparison, when a plant goes to seed, so when a tumor progresses, its seeds are carried in all directions as cancer cells may disseminate throughout the organism, but they can only live and grow if they fall on congenial soil, so can only proliferate in welcoming secondary organs. When looking at the different parts of this analogy, it asks the question of filiation between primary tumor and exited cancer cells, how passive and non-random is the dissemination, what interactions with the new environment, and how can we describe the soil as suitable, favorable, or selective. So from this analogy, which has been cited quite thoroughly in the literature since and still today, our question is to understand how this metaphor has been understood, tested, and reassessed, and more importantly, how it could still be relevant in the light of recent advances on organ tropism and the notion of pre-metastatic niche. As a general view of the chronology since Paget's proposal, so it was first challenged by another researcher so Ewing proposed that the specificity of secondary organ growth was actually only due to which vascular connections existed with the primary tumor. So this was mostly based on mechanical hypothesis. In the few decades after that, Paget was not cited that much, and some experimental data started to be developed mainly based on mechanical hypothesis. And starting in the 1970s, the growing technologies and access to genetic characterizations showed the heterogeneity of primary and metastatic neoplasms and focused a bit more on this idea of organ selectivity, with more or less proposals to test the seed and soil hypothesis. In the 1980s, the idea of mechanical exclusiveness was definitely refuted on a study using peritovenous shunts in ascites for human patients. And a few proposals, a growing number of proposals have been made since to revisit Paget's seed and soil analogy. So as a whole, a big part of my conceptual project is interested in the bibliography of the papers citing Paget since the first paper. So as you can see, starting in the 1930s, the number of citations has mostly grown from the 1980s on, and still today. So we have decided to collect all these papers and analyze them with a certain personal methodology that came out from these readings, with a few different questions on these papers. What type of articles cite Paget? What types of conceptual hypotheses do they propose concerning Paget's analogy and mechanical considerations? How do they more or less precisely describe the notion of specificity of seed and soil? And what may be the biological and clinical implications that are considered by these different papers, in particular concerning the kinetics of cancer progression, the inclusion of systemic factors, and the implications for experimental protocols, diagnosis, and therapy. So from this analysis, we decided to focus on three main concepts that we found would have the most relevance in terms of opening questions for experimental and clinical practice. These are generally how to define the soil at different scales, how to understand the idea of tumor cell or secondary organ predisposition, and where exactly to position the specificity in the process of metastasis. And for that, we are currently working on now analyzing the historical account of reach access, its experimental implications, and the potential clinical perspectives. So this is still work in progress, and I won't detail the whole of the sub-questions, but I'll try to focus on the ones that are the most important for the rest of the presentation. So for the question of defining the soil, we thought maybe different temporal and spatial levels should be considered. We also thought that it was important to focus on the notion of basal soil when thinking about potential predisposition, and how to best characterize soil congeniality or uncongeniality. I will focus here on two main axes, which is assessing basal conditions and with the experimental implication of studying the landscape in pre-cancer conditions with a focus on immunity, and a growing interest on the study of non-metastatic organs in order to ask the question of what is happening in non-affected sites or differently from the affected metastatic tissue. The second main axis is how to best understand the notion of predisposition, and this comes with a strong bias when using certain terms to qualify tumor and host entities. It may have some statistical implications on predictability of metastasis, and shows the importance of scaling up to the organism or patient level as a whole. For this, we would like to focus on the notion of predisposition with a shift to the study of the secondary site and not primary tumor cells only, and the impact on predictability of patient metastasis may have an implication on the importance of using models, animal models, for example, with spontaneous metastasis development. The last one concerns where to position the specificity. So which steps of the process are truly specific? What is the exact role of the pre-metastatic niche? And how to better recognize soil-dependent specificity. For this, the main focus of my project, in particular for the experimental aspect of it, is studying the role of the pre-metastatic niche and asking how early and specific it is by assessing the characteristics of the secondary organ at different time points starting early, and the systemic dimension of pre-metastatic niche signaling that does include the implication of the immune system and encourages us to assess immune presence at different sites and at different times. So this is exactly the question that I want to ask with the more practical aspect of characterizing immune involvement in this seed and soil analogy. By asking the question of how immune actors of cancer progression may or may not necessarily participate to metastasis, contribute to the preparation and specificity of secondary sites, and differentially behave and interact according to the spatial temporal context. For this, I propose to work on a case study, which is investigating the biology of pro-metastatic myeloid-derived suppressor cells in a mouse model of breast cancer, which is known to specifically disseminate to the lungs. So myeloid-derived suppressive cells, and I'm happy here that the previous speaker was able to talk about pro and anti-tumor immunity because this is also important for this topic. So they correspond to a population of myeloid cells derived from the bone marrow that under physiological conditions differentiate into mature granulocytes, macrophages, or dendritic cells, but under certain pathological conditions such as cancer or chronic infections, accumulate at an immature state and may have pro-tumoral activities. These can be subdivided into granulocytic and monocytic populations. So, as I said, under the influence of the tumor context, they are hijacked to become suppressor cells when interacting with anti-tumor immunity, such as T cell function. So they are able to act against the anti-tumor immunity, may also have a role in angiogenesis and extracellular matrix remodeling. and may directly participate to metastasis by interacting with cancer cells and by being present at the future metastatic sites, starting at an early stage, so potentially having a crucial role in the preparation of the pre-metastatic niche. So the main question here is assessing MDSC plasticity and implication in cancer progression, with a first aim of characterizing their presence in different environments. So we are working on a mouse model of breast cancer where cancer cells are injected into the mammary gland, and the metastasis to the lungs is followed at different days after tumor cell injection until endpoint approximately 3 weeks after that. We are using labeled tumor cells that enable us to visualize them by bioluminescence, as we can see here on the primary tumor, and also enables us to detect the presence of cancer cells in different organs when the mice are euthanized. By using this technique, we were able to determine that in our experiment, the onset of metastasis happened between day 11 and day 14. So throughout these different time points, we collect the organs, including the primary tumor and metastatic sites, but also lymphoid organs such as spleen and blood, and a non-metastatic organ, which is in this model, the liver. We want to assess the characteristics of MDSCs at these different time points and in the different organs. So the main observation that we were able to make on this experiment is by analyzing the immune populations by flow cytometry, so labeling the cells with the CD45 marker, and then distinguishing a CD11B, CD11B positive, CD11C negative population, which corresponds to the myeloid compartment, and then focusing on 2 surface markers that are able to discriminate between the granulocytic population in black and the monocytic population in green. When looking at the dynamics of this presence, we realized that it was potentially possible to differentiate between 2 subpopulations of granulocytic cells, G1 in blue here and G2 in red. Indeed, when looking at the presence of these populations throughout time, we realized that at early time points, G2 was most present in the granulocytic compartment, whereas right at the onset of metastasis and until the end, the G1 population may be more abundant, while the monocytic population remained quite low in percentage. So when graphing this in the lungs, we can see this evolution between G1 and G2, which corresponds to the switch between pre and metastatic stages. This was also true when looking at the spleen and the blood, where these myeloid cells circulate. Not so much in the primary tumor, and most importantly, not at all in a non-metastatic organ, which is the liver. So our main questions about this is how to better understand the distinction between these populations. Would G1 be more important in late stages in the secondary organ, whereas G2 may have a role in early steps and PMN formation? And what would be the significance of the monocytic population despite its modest evolution? So this is also a work in progress, and we want to characterize most specifically these cells after purification from the organs to assess their functional specificities, such as immunosuppression on T cells and some metabolic characteristics. I will end this with just 2 short slides of perspectives to come back on the main conclusions concerning the attempted reappraisal of the seed and soil hypothesis, which started with an aim to understand its heritage and update. We have focused, focused on the analysis of Paget's paper and its further citations, which very often cite the paper quite rapidly. But when we dig into the original paper in more detail, we may be able to find concepts that could feed current questions. Indeed, the alternative study that Paget made in his paper, but that has not been much cited since, is to look at different types of affections that were present in the cancer patients other than metastasis. So when using the same cancer patients, Paget also wanted to assess the presence of different abnormalities, such as polypus or cysts in these patients. And here again, a certain specificity was found. So his idea is that with the tendency in women to cancer of the breast, there may be an associated tendency to outgrowths of other kinds and in other parts of the body, which is not observed in women who suffer from cancer of other types of organs. So this idea of associated tendencies and predisposition may be one aspect of what we want to study when we talk about better defining the soil and assessing basal or non-cancer conditions. As a reminder, we aim to better understand secondary organ and tumor cell predisposition and assess the question of which steps are specific in the metastatic process. With this, our main conclusions and claims is that it may be interesting to concentrate our focus early on the pre-metastatic site, both in research and in the clinic, to assess soil potential and basal state activities, in particular the immune landscape in different sites and at different times. And with the input of analyzing the implication of the immune system in metastatic progression, may we propose new terms to this analogy, such as a possible immune fertilization of the soil, as may be the case for MDSCs and the metastatic site in our model. So with this, I thank you very much for your attention, and I thank again my colleagues in Bordeaux, and ready for questions or comments. Thanks a lot.
Thank you very much, Elena. We have a couple of minutes left for discussion. So please again, use the participant tab and raise your hand function.
So can I start, Gregor?
Go ahead, please.
So thank you, Elena. It's great as always. I would like to ask you about your thoughts on the sort of rarity of contralateral metastasis and how it basically sort of, you know, what kind of implications of this is for the seed and soil hypothesis in the current perspective.
Wait, I didn't understand the first part of what you were saying. Can you repeat?
Yeah, so it's that there is the contralateral metastasis, meaning that, you know, from one, for instance, from if you have a primary tumor in breast of a breast cancer, then it's only rarely metastasis. Metastasis in the other breast only very rarely. So I mean, you would expect that, given the seed and soil hypothesis, the best soil for that is precisely, you know, the sort of the same organ as the one in which the primary tumor has arisen. But this is not the case, even though, of course, there is some self-seeding reported. But again, so I'm not sure about the percentage, but it's quite low when it comes to this sort of contralateral metastasis formation. Okay. So I wonder how it affects the seed and soil hypothesis in the current debate.
Okay, yeah, that's an interesting question, actually. I hadn't thought of that precisely. So I guess maybe hypothetically, some possibilities could be the fact that during the course of primary tumor progression, the cells may also evolve and not be as adapted to the primary site as at the beginning, and so maybe more apt to develop in other sites. And then maybe the second aspect of it would be the systemic dimension, whereby from a primary tumor, the signaling to the bone marrow and other organs with the mobilization of immune cells at distant sites may actually be weighing more in the balance of attracting cancer cells to other sites rather than the suitability of the primary soil. But that's only hypothesis, I guess.
If I may just quickly follow up on this. So Do you think that the self-seeding case, so I'm not sure if by self-seeding, the tumor actually seeds the sort of in the neighborhood really of the tissue in which it arose. So if you surgically remove it, for instance, could it be that there is still some tumor microenvironment remaining in that site, so that it's favorable for self-seeding, which is not the case for the parts of the tissue or the others, the contralateral side. So, for instance, the second breast, that would be open to basically accepting the seed and forming the secondary tumor.
Yeah, I guess so. Yeah, self-seeding would be you mean the process of having locally at the primary site the cells leaving but staying in the local milieu, right?
Yeah, so like seeding really still the same origin of the site of the primary tumor.
Yeah, so I guess that would be staying at the level of, well, the organ or a region in the organ, and not so much the other part of the contralateral side. Yeah.
Maybe one more question. I don't see any raised hands.
Can I just one other question concerning the slide? In the eLife paper, there is the tumor intrinsic and extrinsic factors that you mentioned. So can you clarify why exactly is the sort of the interactions with the secondary milieu listed as the intrinsic cause?
Okay, yes, that may not be quite clear. What we tried to do was to, as much as we could, distinguish different types of factors. So I'd say here, maybe some of them are not quite clear, but for example, so you were saying suitable interactions with the secondary milieu?
Yeah, so why is it listed?
So I'd say, yeah, okay, so maybe it should be formulated in terms of specific receptors or ligands on the tumor cells that are specifically binding to the counterpart in the secondary organ, whereas an extrinsic cause would actually be the surrounding environment. Yeah, so maybe that's not quite clear in the figure. Thanks.
Thank you.
All right, let's thank our speaker again.
Thank you very much.
And I think it's